Hello there, my name is Jen Loki, and it is February 23rd, 2023, and today I'm going to be discussing the anatomy and physiology of the retina in the eye. So, I'm going to switch to my screen here. I'm going to be using a few slides I made for the assignment. Let's see here. View slideshow. Okay. All right. So anatomy and physiology of the retina. Okay. So getting to know your eyeball. In order to understand the complexity of what goes on in the retina, it's important to know the other parts of the eyeball, and that is because the light has to get to the retina by passing through other structures in the eyeball. So the first thing that the light passes through is the cornea, which is the sort of the outer covering of your eye. No blood supply, totally transparent. And then it goes through the aqueous humor, colored here in blue. And it enters the pupil, which is the hole in the middle of the iris, or sort of this muscular sphincter that has some pigment on it, where that comes apart in the middle. That's the pupil, and the light goes through that hole. That hole can either be large or small, depending on other neural signals that tell the muscles around it to contract or dilate, depending on whether they're being fired by the sympathetic or the parasympathetic nervous system. But that's a whole different deal. But it does control the amount of light that comes in that gets to the retina. Then it goes through the lens, which is weirdly colored black here, but it should be transparent. And the lens bends the light to then go to the back of the eye, where it deals with the retina, which is this black layer. And the retina, where the optic nerve comes into the retina, is called the optic disc right here. And that optic disc doesn't have any of the structures that we're about to talk about, like the rest of the retina. And the other different portion of the retina, it's worth mentioning, is the fovea right here. So we will talk about that soon. And I didn't mention, after the lens, all of this is filled with vitreous humor. It's like a clear jelly-like substance that the light travels through to get to the retina. Okay, so these are the structures of your eyeball. Okay, so light traveling to the retina must pass through, I'm going to try to keep this presentation under 10 minutes, a bunch of cells before it actually is received by neurons that transduce the light energy into electrical energy. So the order in which the light passes through and maybe gets reflected on some of these cells are ganglion cells, bipolar cells, and through these there are two different types of supporting or glial cells, the amacrine and the horizontal cells too, which you can see here in the pictures. They actually all look like horizontal cells to me in this picture. And then to the photoreceptors. The photoreceptors are turned toward the layer that is deep to them. So the actual receptors themselves are next to the pigment epithelial cells, which is the deepest layer of the retina. And then the photons, the light, the packages of light, are received by the receptors on the surface of these photoreceptor cells. So this is true for everywhere, like I said, except the fovea, where the fovea just has the photoreceptor cells. All of these other cells that possibly could cause light to scatter a little bit and the picture to be fuzzy don't exist in the fovea. This is the center of our field of view of vision, and where we can most sharply focus on things. So there's only this layer of the photoreceptor cells, the rods and cones, which we'll talk about right now, in the fovea. But in the rest of the retina, they have to pass through all of those layers. Okay, the rods and the cones contain three parts. So these are the two different kinds of photoreceptor cells. And rods outnumber cones and except for in the fovea where there's only cones. Cones deal with bright light, so color, and rods deal with low light and black and white. So there's an outer segment. This is where the actual photon gets turned into electrical energy. The inner segment where all of the membrane-bound, or the organelles rather, exist, including mitochondria here, that make the ATP for certain processes to happen, and the nucleus. And then the axon synapses, or terminates rather, in this at the end of this synaptic body. So 3 different chunks of these photoreceptors. Okay. So this is a ridiculously complicated process, but I'm going to try to sum it up. So the phototransduction rather, which is the perpetuation of the signal that starts out as light energy and turns into this electrical energy action potential and goes down the axon and then ends up releasing or changing neurotransmitter release at the end. Rods and cones have very similar structures, so we'll just refer to the one in rods, which is rhodopsin. Rhodopsin is this pigment for low light, and cones also have one for red, blue, and green, and through those can make all of the different colors within the visible spectrum. But in rods, the rhodopsin is comprised of 2 main molecules, and that is opsin and what's the other one? Retinol. Yes. And when light comes in, the retinol and opsin, which are bound together, it changes the shape of retinol, and so it lets go. And this causes a few different things to happen, but in essence, the cation, or calcium and sodium channels that are open when the cell is at rest, close. So without those positive ions coming in, the cell, which these cells rest at about -40 millivolts, not the typical -70, these then hyperpolarize, so they become much more negative because of the lack of sodium and calcium ions, and also potassium ions are allowed to sort of continually leak out, both at rest and during this process. So it hyperpolarizes. This hyperpolarization eventually causes an action potential to be generated, and this moves down the axon and causes a change in the volume of exocytosis of the neurotransmitter glutamate. So normally at rest, glutamate is tonically released, so all the time, just always. But when light is in this formula, it changes. So it can decrease. So here on the right, these 2 pairs, this pair of neurons, the glutamate has stopped. And so it can do this. It can stop, and it can kind of do anywhere in between, depending on, I guess, the number of repeated action potentials. And then this glutamate can have an excitatory or inhibitory effect on the bipolar cell, which is the secondary neuron that this message is being sent to. So now the message, I guess we'll say, is moving from the deep layers out. Remember, we first talked about the light entering and going to the retina and having to get through these layers of cells, but now it's kind of doing the opposite. It's going from the photoreception at the sort of the top of the epithelial pigment cells, where that's where the photoreceptors on a separate cell, but right next to that layer, take in the light, and now the light is kind of moving through these other layers that are superficial as we move out, more and more superficial. So many photoreceptors converge onto a single bipolar cell. So we could have many different photoreceptors doing different things with their glutamate that then bind to these different types of receptors on the secondary bipolar cell. And then the message is then sent through the bipolar cell and then onto a ganglion cell. So, many bipolar cells can converge on ganglion cells. So when you think about the number of photoreceptor cells to ganglion cells, there are many more of those photoreceptor cells than there are ganglion cells. The ganglion cell body passes this neural impulse on, but the axons of the ganglia or of the ganglion cells are what make up in bundles that optic nerve. I'm getting ahead of myself. So it then heads down the axons, which are the optic nerve, hits the optic chiasm, I don't really know how to pronounce that, the optic tract, the thalamus, and then eventually the visual cortex or the occipital lobe in the brain, which deals with those messages, and then can send it back out through efferent, I don't know how to pronounce that either, neurons that might change the musculature or do something else once that neural impulse of the light energy has hit the central nervous system. It can then be sent back out into the periphery, but in efferent cells. I think that's it. And my reference was simply our textbook that had plenty of information in detail. Let's see here. How are we doing for time? Okay, 11:30. Not terrible. And then my study guide is a sheet that sort of a structured notes. When I was a teacher, I did this all the time, actually. I would scaffold the notes in the order of which I would say things and then have kids like fill in the questions as they go. It's a little less overwhelming than just having to take notes. So I think that's it.